Sarah will probably be able to introduce herself much better than I do, but
yeah, but for those who haven't met me, my name is Sarah Ramdeen. I'm one of the curators at SESAR, the System for Earth Sample Registration. My background is in a mixture of geology and information and library science. So I've got an undergraduate degree in geology and worked collecting cores out in the field before transitioning into more of the data management and data curation side of things.
Thanks, Sarah. You give the go, Jeff.
I think we're ready, Sarah. I think we've probably got who we're going to get. So thank you for joining us today. We appreciate you being willing to do one of these webinars, and welcome to everybody who's here. I'm excited to hear more about this, and you should take it away. And Sarah, you can decide if you want people to interrupt with questions in between, or if you want people to save questions for the end. That's totally up to you. Okay.
Yeah. So as you mentioned, today we're going to talk about registering samples with SESAR. And let's just go over the agenda and then we can decide. First, we're going to go into some of the motivations behind sample management. We're going to introduce you to the concept of the IGSN. Then I'm going to give you a brief introduction to SESAR, followed by a demo where I'll walk through some of the processes of using SESAR. And then we'll have time for questions. I'm fine with asking questions in between, but Kirsten is going to kick us off with the motivations and introduction to IGSN. So are you all right with questions too, Kirsten, while you're talking?
Yeah, I mean, I would consider these real discussion sessions, and, you know, we're going to present stuff. And when you have a question, best time is to ask right away, because others might benefit also just from, you know, learning more of the background or clarifying if something isn't as clear as needed. Yeah, okay, why don't you get to the next slide, Sarah? So I just wanted to introduce this a little bit. Why are we talking about samples actually, you know, as part of the data management team? And I think it has become pretty obvious for the critical zone network here that, you know, nearly every one of the clusters is collecting samples, and it demonstrates how fundamental the process of collecting material out in the field is to the research that we're conducting, and you know, all the analytical work and experimental work that is done in the labs. So samples have become part of the digital research data ecosystem in some way, and that is because if you don't have information about the actual samples available when you report data that were generated on the samples, those data don't make a lot of sense to people and can't be you know, reused or reproduced in the same manner or in the necessary manner. So all these discussions about fair data, making data findable, accessible, interoperable, and reusable do in many ways also apply to samples. You know, and as I said, it's for the same reasons as we talk about fair data, that science needs to be open, transparent, reproducible, and that there is a lot of resources going into generating the samples or generating data, and therefore we want to make these available for further reuse. For samples, there's also that aspect that if a sample gets further analyzed in the future, you're actually gaining a lot, you know, and that you're adding knowledge to existing knowledge. And so sharing samples, preserving samples, and making them accessible is incredibly important. So, but there's actually direct benefit for researchers and technical people, students, whoever, who are working with samples. You can make your work much more efficiently if you follow best practices. And that pertains to tracking samples, sharing them, and later also citing samples in publications or in data sets, and making sure that data get linked properly to the samples, and that people who have collected the samples and who curate the samples do get credit for their work. So I talked about the scientific impact of the samples already. So Sarah, next one. This is just an example, and I think it's actually a really important aspect for work of the critical zone clusters and the type of samples that they collect. This was an article about the repository of global river waters that has been set up at Woods Hole. And, you know, when you think about it, when you collect a sample out in the field, you're basically taking a snapshot of nature that can never be recollected again. So in this case, it was, you know, somebody saying, you know, maybe 10 years from now, you want a sample from the Fraser River water in 2010, where do you go, where do you get it, you know, and people really need these, you know, like saying, I've got a lot, I would give a lot for a 1930 or 1960 water sample from the Mississippi, you know, it's gone. So these are reasons why we want to preserve samples and you know, document them properly so they can be reused, but also so they can be found and accessed online. Next slide. But there are, you know, other aspects, and those pertain to NSF's data policy. Data management plans are supposed to include information about what will happen with your samples. And therefore, you know, if you have a repository where you need to send your samples at the end of the project, it's important to have them documented from the beginning on, and again, making work efficient. So at the end of the project, you don't say, oh my goodness, where do I have all the information, the lat long where the sample was collected and so on. So it's sample management is important for compliance with these type of data policies. And I think also it is important, while this work of the critical zone network is going on, that there are opportunities for sharing samples, because as I said before, you know, analyzing samples that somebody else has already worked on will add knowledge and add more opportunities to correlate different properties of the samples with each other that come from different perspectives, be it isotopes or be it you know, content of organics and so on. Next slide. The other aspect is that increasingly journals will request unique identifiers for samples, so they properly referenced, and information about the samples. And, you know, Sarah will go into talking about the IGSN and how, you know, if you have an IGSN for your sample, and put that into the publication, there is about that application will always link to the landing page of that data set, be it in HydroShare, be it in EarthChem, and so on. So the compliance with journal policies also increasingly is an issue that proper sample management helps with. And I think with this, I can hand over to Sarah, unless there is a question right now already. If not, then, yeah.
Did you want to go through the rest of your slides, or did you want me to walk through them?
I thought this could be already for you, but I can give you whatever you want. Go ahead. I think, you know, this gets into what is required in proper sample management.
Yeah, so we've probably all heard the concept of FAIR before, and that there's a lot of prescribed recommendations on what would be needed to have FAIR research outputs. And here is a list of some of those things, like unique persistent identifiers, making metadata discoverable. And one of the things we want to highlight is that, if I can make it advance, there we go, is that by using SESAR, you address a number of these facets. So SESAR provides the IGSN as a persistent identifier. We have these metadata landing pages that are persistently maintained by SESAR, but updated by you, the user. And then we have a different other various aspects of this concept of FAIR that we address as well. And again, we're happy to answer any questions about that, but we might address some of that as we get more into how SESAR works. And we've mentioned it, but we haven't really gone into what it is. So the IGSN is the globally unique persistent identifier for samples. So one common way to think about this is that, let's say you're out in the field, you collect a sample and you name it IC1. Well, someone else might go out into the field and name their same sample the same thing. And once you get to that publication or sharing stage, how do you tell the difference between my sample and your sample when we both named it the same thing? And also, as you move through a lab, how do you make sure that the name is consistently applied and you know which sample is being taken care of as additional analysis is done or other metadata is added to the sample? And so that's where an IGSN comes in. It's an unambiguous identifier for the sample that uniquely identifies it and also has this additional aspect of being both a persistent identifier and resolvable. As Kirsten mentioned earlier, it's like a DOI where you put the string into your browser and it will lead you to the landing page for that sample, that metadata profile for the sample. And IGSNs are assigned to both physical objects like rock specimens, water samples, sediment cores, as well as related sampling features like sites, stations, stratigraphic sections. And really, IGSNs are help used to do some of these best practices. And some of these things that we've already sort of been dancing around, the ability to find or locate a sample out in the wild, from publication to the archive where it's housed, and to access it, to then be able to either ask for additional information about the sample or to actually be able to go visit and look at the sample. It helps link it between different resources, between the data set, the researcher, the article, maybe the funding agency that funded the project. And it also allows you to cite the sample as well. But by using this IGSN, you connect this whole network. And as you can see, now samples are part of this broader knowledge graph related to research data. So before we transition into what is SESAR, do we have any questions, as Kirsten mentioned, about some of this background and motivation? Okay, seeing none, I'm just going to hop right into it. So SESAR is a community platform that helps make samples more discoverable, accessible, and reusable. And it also connects samples with the knowledge ecosystem where they're derived. So SESAR offers a wide range of services to support scientists along the life cycle of a sample. We provide a dashboard to support metadata management, a public catalog of user-contributed sample metadata to support discovery and access, and then we provide IGSN registration to support unique identification and tracking of the sample as they move from field to lab to publication. So users manage their sample metadata in our dashboard, which is called My SESAR. And during the demo, I'll walk you through how this works. But My SESAR is a private workspace which allows users to register their sample metadata, and this can be done through an individual registration in the My SESAR interface or through bulk registration. And it also is where we issue the IGSNs. So within My SESAR, users can view and edit their submitted metadata. My SESAR also has a number of tools to support metadata management, such as adding images or files to the sample profile. We have features that let users manage permissions, such as access to sample metadata, which you might embargo them and restrict access to it. You might give others permission to help edit the sample metadata. And then you can also transfer your samples to another person. So let's say you submit your samples to an archive, you can actually transfer the metadata records to that archive as well. And then My SESAR also has tools that allow users to create groups and then export the metadata for the samples in those groups into the set of EarthChem library templates. So as we've mentioned, SESAR maintains the sample profile pages for every registered sample. These samples contain all the metadata submitted to SESAR, and that could also include images like the sample here on the slide. It could have other associated files, and then there's other information included in the record as well as perhaps the description of the sample, where it came from, the geographic location, where the samples are currently stored, the scientist who collected the samples, and so on. When a sample is registered in SESAR, it's issued an IGSN. And as we've mentioned, they're resolvable and they lead to this landing page, which we're previewing here on the slide. Let's see. And so the registrant owns that metadata and can update it at any point in time. SESAR just ensures that it is a persistent page and that it will always be there. We also have a public catalog, which supports allows researchers to find existing samples that might be relevant to their research. They can use the search catalog using geospatial information, sample type, sample classifications, and other criteria to search within our collections. And then from the results, the users can view the sample results on a map. They can download the sample metadata from their search results, and then they can also look at the individual sample profiles. And that's a big part of SESAR, is this discoverability and access. And we'll touch on that again when we talk about some of the metadata you might submit into SESAR. And so I'll pause at this point again for questions. And then if there aren't any, I'm going to go through a demo of SESAR. I'm going to show you how to set up an account and some of the different facets of account management, and then go through the process of showing you how to create a template for submitting bulk registration, and then show you how to actually register the samples. I can remember how to get out of the presentation mode. Nope. Sorry. I'm instead showing you all the slides. Just a second. Let me exit out of share and then close the presentation, and then I'll move into the demo. There we go. Sorry about that. Okay. So to enter my SESAR, if you go to the SESAR homepage, which is geosamples.org, we have a My SESAR button here at the top, and it will lead you into the login feature. And I'm going to move over to our test interface at this point, because I'm going to be doing just some fake registrations and I don't want to use them on the live system. We recommend, we have two different methods for creating an account. There's using your ORCID, and then we also have another in-house method called a GeoPass ID. And we're currently encouraging users to use ORCID, but there are some reasons why you might need to use a GeoPass. That could be where if you're creating an institutional account, which can't be not tied to a unique person like an ORCID is, and also to use our web services or API processes for registering samples, you would need a GeoPass as well. I'm not going to go into creating a GeoPass right now. I'm just going to focus on ORCID, but if you have questions about GeoPass, we can talk about that either in the questions or in a one-on-one session. So here I'm clicking to log in with ORCID, and I should already be logged in. Oops, I am not, so unfortunately you're going to have to see me. I'm going to stop sharing for a second so I can put the password in. One of those things. But when you first sign in with your ORCID, it's going to ask you to add us as a trusted account. And that's a one-time thing where you shouldn't have to do it again. And that just allows SESAR access to some very basic information like your user, your account name and things like that. So let me go back to sharing my screen now with the passwords in. Okay, so here it is, it's asking to access your ORCID record through SESAR. And so we'll authorize that. And that will only happen the first time you log in. And here, oops, I must have gone to the wrong one because I was supposed to go to create a new account. Sorry. I must have accidentally created an account earlier when I was going through some of this demo practice. But normally, once you created that account, it would lead you to this page and it would land on your account profile page. And you'd be required to finish filling out your account profile before you can move into using your account. And then it would also prompt you to create a user code. So for SESAR, we do some account management through what's called a user code. It's a 5-digit character starting with IE that might represent you as a researcher. This is your user code, and all samples registered in SESAR start with that user code. Or it might be project-based or lab-based, and it's a shared code that you'd use amongst a team of researchers. So this case, I'm just going to make something up, which I'll end up deleting from the system later. And as you can see, it's added it, and you would need to do that before you could do anything within my SESAR. But as I mentioned, if you create something like a user code for a lab, we have a process where you can share permissions. So here under my account management, I can say, I want to give someone else access where they can only read the samples, or perhaps I want them to be able to actually read, write, and edit samples. And you can set that to be for a certain date range, and then you would either enter their GeoPass or their ORCID ID, and then give them permissions to use your user code. And if you have questions about how to set up user codes for your lab, again, that's something we can talk about during the discussion or in some one-on-one consultation on what works best for your lab and your situation. So now that we're in my SESAR, I just want to show you a little bit around. Once you create your account, you'll normally land on this homepage when you log in, which is where you can get to different areas where you can see your samples that you currently have registered. And actually, I'm going to switch accounts because I want to show you some test materials that I already have lined up within SESAR. So let me just switch accounts. So here you can see your samples that under my sample that you already have registered, and you can edit them individually if you wanted to. You also have the option to do some other things such as transfer ownership of samples to someone else and some other tools. The main thing I want to show you today, though, is how to register samples. So we have 3 methods of registration. The first is our individual sample registration, and that's just going through the MySESAR interface and registering a sample using our GUI interface with drop-down menus and registering a sample one at a time. It's a very manual process. You can see which fields are required, and it will walk you through it. Sorry, the Zoom menu is covering some of the buttons here. All right, I can't go on to the next screen. There we go. Sorry, Zoom was covering things. And it will ask you some questions and walk you through the registration. That's often time consuming. It's easy if you're doing like 1 sample, but if you're doing 30 samples, you're not going to want to use that method. And the main process people often use is our batch registration. So to get started with the batch registration, you would first create a batch template file. And one thing to note about this is that when you register samples in SESAR, you can register 1 object type per batch. So let's say you were registering some water samples as well as some rock samples. You couldn't put them in the same batch registration. You'd have to do them based on object type. And so when creating a batch, that's the first thing it asks you. So which user code are you using? Because you might have more than one. And then what object type are you registering the samples with? After that, it will then prompt you to select which metadata fields that you'd like to see in your batch template. This lists all of our possible metadata fields. And if you mouse over the fields, it will give you a definition of them. Once you create your template and hit submit to create template, we'll actually download 2 files. One is our quick guide, which is similar to that mouse over. It has a listing of every field possible in SESAR with an example of what the data might look like, a definition of the metadata field, and then some additional information in context. For example, for some of our fields, there are controlled lists that you need to use. Others might have suggested lists or recommendations on how to best use the field. You'll also get a template, a blank template that has the object type you've selected, your user code, which are these main header informations required in a batch template, and then the metadata fields that you selected that you'd like to register. So let me go back to the slides real quick. Because one thing I wanted to talk about was, so when you create these batches, you'll want to then spend some time mapping your metadata to the SESAR metadata fields. And we can set up times to talk through you and walk you through this process of saying, well, SESAR has this field, what's the best fit to my metadata and how might we adjust that? And ultimately what you'll end up doing, I'm sorry, I don't want to go into present mode and then get stuck in it again. But ultimately what you'll end up doing is then transforming your native metadata into our templates. And let me move back over to the templates to show you an example of one. So in the end, once it's full of metadata, here's an example of a completed template. It's got the sample name, these first few fields are blank, and then there's additional metadata about the samples themselves. And
Sarah, are you showing something different? I'm still seeing your PowerPoint slide.
Oh, no. Let me try making sure it's sharing my full screen. Is it showing the template?
Yes, yep. Mm-hmm.
And just to check earlier, when I was talking, did it show you the quick registration guide?
I have not seen this screen that you're
No, it
showing
didn't,
right now
sir.
yet.
Okay, I apologize for that. Earlier when the files, just a step back, when you download the batch template after you create it, it downloads this quick guide, which, as I mentioned, has the definitions, and then also a blank template with the metadata fields you selected and the required header information for the batches. Sorry, just to let you see what it looks like, and then show you what one now looks like, the metadata added to it. Okay, and now I'm going to go back into my SESAR and let me know if you can see. Do you see the my SESAR screen?
Yes.
Okay. So now you've got your template worked out and you've got your metadata as you want it. You would then go into my SESAR and upload the batch for registration. So I'm going to show you first a bad file, and then I'll show you a good file because I want to do this for an important reason. So I've uploaded a batch. SESAR has built into it some validation checks. And so automatically when you upload it, it's going to look for and say, this field has controlled metadata. Are you using a term from the controlled list? Or perhaps this field is supposed to have a date in it. Are you putting in numbers or are you putting in letters? So we have some preliminary validations that happen when you submit to SESAR. And as you can see at the top, it's telling you what the error is. And then the field that has the problem is highlighted in red. So you can do some troubleshooting. And if you go through this validation and you have an error, it allows you to then cancel the upload and upload a new file instead. So we'll go back out and now I'm going to add a good batch. And so with this one, I don't see any error messages, but I'm able to still preview my metadata before it gets submitted and do a simple check to say, okay, our field's going to the right way, just as a check, like is sub object going into the right column and so on. And then you can scroll through and check your metadata. You should also be able to side scroll, there you go, and look across all the fields that you've submitted. And then you'll click register now to upload your file. And once it's submitted, you'll get the successful submission screen. At that point, the submission is sent to the SESAR curator, and we'll go into the system and we'll download your submission, and we'll do an additional check on it there. We'll see if, does your object type make sense for the sample metadata you've submitted? Are you using all the fields that we recommend, such as like location or archive? Like some of those are not necessarily required, but they're encouraged, and so we'll do a check on that. And then once it's processed, we'll send you back your submission file with the IGSNs added to the file in that column that was previously blank. So in this file here, column B will now have the IGSNs listed in it. And then you'll also see, if you went to my samples, you'll be able to see your samples registered there. That turnaround is typically, we try to do that within a day of submission, depending on the size of the submission. We have gotten submissions that have like 4,000 records. That might take us a little bit more time to just spot check, but typically it's about a day turnaround. Let's see. Just want to keep an eye on the time. I have a few other things I could show you. We do support pre-registration of samples. So let's say you're going to go out into the field and collect samples, but you want to be able to print labels for your sample collection devices in advance. So maybe like the vials for the water or the rock boxes or bags. We do allow you to do pre-registration where you would register a template with mostly blank information and then later come back and update that file. I can show you what it looks like to update a file if you'd like. I'm just trying to think. I do want to keep some time for questions. Let me just look at my notes too for what else to demo. Okay. So in this case, just to show you here, when I registered these samples, we could go through that, but I won't be able to show you the curator side of things. But I did already register previously. Where did they go? I registered some test samples that are in here, and I have another batch that I can show you where it's the batch update. Where now here I've got my file back. It has the IGSN in it. I'm replacing that pre-register with now additional information, and then I've added things such as location and elevation. So then when I go back into my SESAR, in that same area where you register your samples, you can also update them. And so this update feature is important because it's not just for pre-registered samples, but at any point in time, if you have additional metadata or information about your samples that you want to add to the records, you can do them individually through looking at the page and editing a single file, or you can do them in batch if you're applying a bunch of information to a bunch of samples. And it is pretty straightforward. So let's see. Got my file, I go through the same upload process. It does the same sort of validation checks that it does on a batch upload for an initial upload. But when I hit update now, it's automatically updating them live. It doesn't go through the curator, it automatically updates the metadata. And then as I mentioned, the other method to edit a sample is in my samples to identify the sample you're looking for, and then you can click edit and you can edit the individual record itself. And this includes adding URLs or adding files. And once ready, you just hit submit, and again, it automatically updates the record at that time. Okay, let's go back to the slides. So we wanted to give time for questions, but I did have a few points I just wanted to address first. So as I mentioned, SESAR supports pre-registration of samples. So before collection, again, to help with that process of either label creation or to assist with metadata capture. You can register a sample that's been destroyed or will be destroyed, because that is one thing to be able to preserve the metadata, knowing that, all right, I'm going to run a destructive analysis, but I still want to be able to track that result and tie it back to a sample that existed. And then also sample metadata can be kept private for up to 2 years following NSF guidelines after the registration date. And so that's a field called release date, where you would set it to a time in the future, and we cap that at 2 years. It is adjustable if needed for variation, but we encourage you to release them publicly within that first 2 years. The IGSN is still resolvable at that point, but it will lead to a landing page that says the sample is private and will not share any of the metadata about it. And then just a few question points I want to kick out there. I'll save those for first to see if there's any questions. Sorry, I know that was a lot of information all at once.
And I just wanted to add one piece of information, as you also mentioned sites and so on, that you can link related samples and sites, subsamples from a sample, so that you can track that whole processing and analysis of subsamples and so on, or you could register a site and then link through the metadata or the samples collected at that site to it, and build these, yeah, kind of genealogy of samples. I mean, one suggestion I would also have is when, you know, you start registering samples and using SESAR, maybe we have another session at a later time just for questions that come up, or so, you know, that, or just, I mean, bring them to the help sessions that we have every other week. Because
Sarah, I have a couple questions.
Yeah, okay, go ahead.
It looked to me like the metadata for samples is really quite flexible. So 2 questions related to that. Are there instances where people come to SESAR and try to register a sample, but they don't find like a specific sample type, or I think you called it an object type that works for what they're doing? And then the related question is obviously, what if you look down through that big long list of metadata elements and you don't see things that you actually really want? How do you sort that out?
Yeah, and that was some of what I sort of knew we might need to discuss with this community. Is there's 2 levels to that. One is that we might help you find the right answer because we might already have it, but maybe perhaps you're not thinking of it in the same context for discovery. The other thing as well is that for some communities, we do work with them to add additional terms to our controlled vocabularies. So for example, we were working with the TEFRA group to add TEFRA as a sample type. We've also, we're working with the ICE Corps facilities to add some material types related to different types of ice cores and other variations like that, where the metadata hasn't been needed yet. We haven't been working with those communities, and so we will add those terms to our controlled lists. We will often ask when that process happens is that those terms are vetted by the community. So it's not just an individual lab saying, hey, can you add our esoteric way that we call the sample here, but something that's more shared and communal, again, thinking about it from the perspective of someone else coming in and discovering it and accessing it later. So we'll ask either that you shared valid sources that back up your term, or that it comes from having been community vetted as well.
And what are there like minimum metadata that are required? So that was a big long list of metadata elements, and I can check them on and check them off. So are there things that I absolutely have to have? What controls? I mean, I realize you're looking at all of these as people are sending them in, but what controls whether I'm, selecting all of these metadata fields that you think are important versus what I might think is the minimum set of metadata that I can give you to get my IGSN?
Because we support that pre-registration, the minimum metadata is actually very limited. It includes the object type and the sample name. And I think those are the two only required fields, and the user code. But at that point, that's because we're expecting that some people might need to register something before going out into the field. When we do the curatorial review, that's where some of that maybe a little bit more encouraged metadata fields might get pointed out if they're not obvious in the submission. And for that, we generally like to see a location. You don't necessarily have to have latitude and longitude because not all samples have that information, but something about where the sample came from or something that gives it context, locality, a state, something. And then we'll also like to see contact information, and that might be for the scientist who collected the sample, or it might be the repository where the sample is now housed, so that someone knows how to get back in contact to say, oh, I found this. I wanna see the sample and know where it's at, or how to get more information about it. I'm trying to think. We have it depending on the sample type too. That's where, again, we might recommend some other additional fields. But Kirsten, do you have any?
Yeah, I mean, there is this perspective of SESAR being quite, or the samples being quite diverse that come to us. And so we had in the very early days, lots of museum samples that did not have that long, so we couldn't really insist on that, right? We have sample types, especially something like a dredge that is a mixture of different materials, where you can't really insist on having a single material. You would have to have like composites and so on. So sure, you can build all that, but we never had the resources to have, all these controls by each object type and so on. But with respect, what I just wanted to add is SESAR is entering actually a phase where it's going to change, and its metadata schema will become a lot more flexible, so that different profiles for different sample types will become much easier to implement than it is now, where it's a fairly static, rigid metadata schema. We are part of a project that's called the Internet of Samples that is building software packages to ensure an easy to implement sample management or sample metadata management systems that all talk to a central system. It's called the iSample Central and iSamples in a Box, but I don't even want to go into the details, right? But the central registry in that iSamples system will allow users to pull metadata profiles that they need. So if you say, I have a soil sample and that needs, to have this particular set of metadata, then you can use that. And there is, a lot more flexibility in it than it's meant to be, applicable in the broadest sense. I mean, we have participants from archaeology in this project to come with their very specific metadata requirements for archaeological artifacts and so on. Right now, we're just trying to squeeze things into an existing database, but that will change. So, you know, again, as Sarah said, it is critical, I think, now for the cluster participants to think about, you know, are there specific metadata, specific terminology that should be implemented for the CZNet so that, you know, searches may work. I mean, one thing we want to talk about briefly, I think today is, you know, how should a search for samples that are particular to the CZNet function? Do we need that? You know, and depending on how you want to find samples, that determines which metadata need to be available and what terminology will be used for that.
Can I ask a question about that, Kirsten? If there are specific metadata fields that we want to have for CZNet that sort of, I won't say uniquely, but sort of specify this is a CZNet sample, how do those metadata elements get exposed to discovery? Like, is the discovery interface savvy enough to pick up on those sort of custom metadata fields, or how does that work?
That's also in change right now. So it will be much easier. We are in the process of basically creating an index, an Elasticsearch index of the whole database, and build a much more user-friendly search of these samples. iSamples itself has already built a very cool search interface, but it's only in a prototype right now. But that will also make all samples from SESAR available. But right now, it's what we've done is, for example, if you have, you know, you want to tag samples by cluster or by, I mean, the site is pretty obvious how you would do that, you know, that fits into metadata that are already there. But, you know, we have used for the critical zone observatories, we've used the field for field program, which, you know, in the marine world, it's the cruises. We've used field program to name specific critical zone observatories so you could find them and so on. So yeah, it's been a bit of a yeah, pushing something into an existing structure, but that will hopefully be much easier to accommodate new metadata.
And to answer those questions too, is that's what we'll work with. We can work with the different clusters on their needs. And we've done that with different communities where we say, this is the recommended field where we think your metadata information should go. So the best example I can think of is here is that we were working with the Alaska Volcano Observatory. They have volcano information, and we mapped it to our primary physiographical feature field. And so we can work with you to say, maybe we don't necessarily have the exact fit field, but here is the best fit that will also make it discoverable through our system, and knowing which ones are indexed and searched currently as well. And then some of the other things that we've done, and I didn't get into it here because I wasn't sure how much had been talked about or discussed, is we've also worked with what we'll call different communities where we will develop a community recommended profile. So right now we're working with the TEFRA community I mentioned earlier, and they've created a SESAR template where they said, these are the fields we would like to see if you're submitting TEFRA samples. And then they could tell other people in the TEFRA field, use this version of the SESAR template where we've already identified which recommended SESAR metadata fields you should include, and then a recommended mapping between how the TEFRA community talks about their metadata. And so we went through a process with them to work on that. And we can do those with some of the different clusters as well. And with both levels, thinking about the cluster and then also thinking up to CZNet as well, if there's things that should come from the top level versus the individual domain levels.
I would actually say, you know, it's rather than doing this by cluster, it would be good to go back to our metrics that, you know, Jeff put together of all the data types and sample types that were sort of listed by the individual clusters, and say, okay, let's this time think about, you know, dust samples, or let's think about soil samples, or, you know, whatever other. We had a whole bunch of wet water samples. We have the cores in the what's the cluster called? Deep regolith? No.
Yeah, the bedrock cluster.
Bedrock.
The bedrock cluster, yeah.
Bedrock cluster, you know, they have cores. So there's obviously, we already have profiles, metadata profiles for cores, but that would be the recommendation. We also should mention that we've worked pretty extensively with the ESS dive group at DOE on developing metadata profiles for soil samples, right? With soil samples.
And also some other sample types, they have like filters and filtrates, I think.
So the folks who are on the call, what are the sample types that you're working with? Nobody is working with samples?
At CNET, we have SolCore, and I think they already got some IGS numbers registered for them.
Yeah, I know some came in. Great. Yes.
And we're happy to have one-on-one meetings with you too to talk about some of this more nitty-gritty things of like how to set up an account, how to determine a workflow for when you have multiple people working on the same samples or in the same lab, determining best practices for creating a user code. And we also have some recommendations for that on our website too, under resources for institutions and resources for researchers.
So let me just come back to that question that we asked earlier about searching for samples. So, I mean, I'm not the researcher working within the cluster network on samples and on analytical work, but what do you see? Is there a benefit for, you know, when the Hub CI team looks into discovery of CZNet data, to have a component where you can discover CZNet samples to say, well, what type of samples has this other group collected, or across the whole network? Are there other people who have collected this particular type of rock, of sample, of soil, or of water? I mean, the IGSN really applies to all these different types of samples. It's not just for solid materials, it's for water, it's for gas, it's can be leaves. We have, yeah, from the old critical zone observatories, they registered organic components, biological components of the soil and so on. So any one of these object types can and should be registered. Or maybe it's worth discussion within the individual groups, right?
I had one more question in my list. Sorry if we still have time for a question.
Yeah.
You demonstrated metadata update. Either you can update the metadata for an individual sample, or you can do, I think, batch update. You said we can do batch updates. Are there any restrictions on the metadata update? Like, because presumably the IGSN already exists when you're updating the metadata. Do you have any restrictions on what they can update versus what they can't update?
You cannot update the IGSN. So once that's set. You can go ahead and change the object type though. So for example, if I'm pre-registering and I don't know what the object is, you can just put something as a placeholder, and you can update that. The user code is tied to that same, and just to check, you are seeing Excel right now, right?
Correct.
Yeah.
Okay. So I hate to do that again. Yeah, so as far as I believe those are the only, and release date, I believe those are the only 3 fields that you cannot necessarily change. And when I say you can't change release date, you can set it further in the future, but if the date has already passed, you can't make the samples private again. So in that case, you can't change it. There may be some complications as far as, where is it? We have fields for related URLs, and that's a one-to-many relationship where you can have up to 5 related URLs for a sample record. But when you do the update process, you might overwrite the fields if you put metadata in there. So that's something where you'll want to be careful as far as order of operations. What's already in there, and are you adding, when you update, you add a new record rather than overwriting what's already there. So you'll have to be careful about thinking about, am I removing what's there or am I adding information? And in this case, when you do the update, you're only adding information as opposed to removing the existing record. So you'd have to manually delete that if you're trying to remove perhaps maybe a DOI that you put in incorrectly. The other fields, I believe everything else is fine to be edited. It's just one thing to keep in mind with the update. I should have, because I was pre-registering, it didn't matter. I listed all these fields and I left them blank. But if I use the same file and I upload it again, and let's say I had manually edited a record and added some of this information, if I included the column here and left it blank, it's going to overwrite what I manually entered through my SESAR. It's going to update it with this blank cell that's here in the batch template. So if there's a field that I've already edited and I don't want to change, you would want to not include that column in the batch template. So some of it's just, it's not necessarily about what SESAR doesn't let you from a conceptual process. It's more about just the logistics of how to use the update process. And we do have a tutorial on our website that walks through some of these do's and don'ts. Let me see. In our resource section, we've got a number of tutorials, and we've got one that will walk you through creating a batch template, as I showed here, in a little bit more detail, and then also this batch update, and that has a little bit more information about that. With going into that, careful of don't overwrite data you don't mean to.
Yeah. Makes sense.
I just wanted to end with one important statement. You know, registering samples is the first step, obviously, but then you actually need to take advantage of the IGSN, and specifically, it is important that the IGSN makes it into the publication in the end, because that's where, you know, when the data are reported by IGSN, I mean, reference to a specific sample, but with an IGSN, that's how in the future researchers will be able to link new data to that given sample based on the IGSN. As Sarah said, if we all name our sample IC1, that's fine. You can do that, but you need the IGSN next to it so that it actually is uniquely identified. And new data can be linked to it with that same, via that same unique identifier.
Yeah, and I had this as a backup, so I'll just put it up. It doesn't exactly address what Kirsten's saying, but it's the idea of using the IGSN as that linking either in different systems or in that center one. You can see it's in the publication, and you might link it to other related identifiers as well. I think also we had that earlier diagram, might book this one, might also better display that too.
Yeah.
Well, I'm going to go back to this last slide here, just to close things out, to say that, and this link is also available on our website in our resources section. If you follow the bit.ly link at the top here, it's a Prezi that goes into a little bit more depth and more examples of what I've shown today on how to use SESAR. And it also has links where it shows you how to use EarthChem and Astromaterial or related systems. And then also, if you have any questions about any of this, or if you want to set up a time to talk one-on-one about anything, either metadata mapping, account creation management, anything at all, feel free to email us at info@geosamples.org. And then also one of our team members will be at these weekly help desk sessions. So if I'm not on the call, Lucy could always pull me in if needed as well.
Great, thank you for that presentation and the great information. I learned a lot today, so thank you for answering my questions. If there's no other questions for Sarah, we will wrap it up for today. We're out of time, but if you do have questions, please feel free to bring them to our next data help session, which will be next week. And then the following week, we'll be having another session. We'll be talking about the Zenodo repository, which we're offering sort of as a catch-all third option for submitting data. And definitely not the only third option, but something we can talk about and talk about where we might be able to use it. So we'll see you all next week.
Thank you, everyone.
Thanks. Bye.
Thanks, Sarah. See ya.
Bye.